So I'm here talking with my colleague, who actually is a experienced and highly trained crystallographer from Johns Hopkins, who has done a great deal of crystallography, and I've asked him to take a look at this structure with me to help explain the more nuanced things, which I, as just your average biophysicist, don't know. And so what we're looking at here for the magenta, you can see this is the GLP1 receptor. So the GLP1 receptor, that is a transmembrane receptor. Part of the GLP1 receptor is going to be buried within the cell membrane. The purple or bluish area is the semaglutide peptide backbone. So this is the semaglutide peptide backbone. And finally, these funny looking structures here, you can just ignore those, but just because you'll wonder what the heck is that. These are the chemicals, ions in the water, and for the purpose of our conversation, we're not going to talk about why those are important, so let's just try to ignore those. To orient ourselves, we can see here that, remember that the semaglutide has the first 7-ish of amino acids removed, and what they have crystallized here and the structure they've analyzed starts at position 10. If you'd walk through it, you would see that as you go through this wonderful little helix, that the numbers go up, right? 8, 19, 20, 21, and so forth, till we get to the very end at 37. So this is our semaglutide. And it is, remember, when you take the semaglutide, it binds to the receptor, and this is where all the magic happens. The receptor transmits its message. It's kind of like its own little Twitter or X. It gets the message and it sends it throughout the body. And so a cell membrane receptor has a couple of motifs that I'm going to hand it over to our crystallographer colleague here, where some of these areas, which are called beta sheets, are buried within the membrane and stabilize the transmembrane protein. And these structures that are going to be described in a moment are what's critical for that. But you can also see there's these helixes that the transmembrane protein has too. And it appears, at least to the layman's eye, that maybe that's what's associating, although I'm not seeing any bonds. That's not what you do with this anyway. So with that, I will hand it back to you. What are those yellow things in there, in the purple structure? Because the blue doesn't seem to have any of those yellow areas.
Those are the disulfide bonds. So let's look at this. If you imagine a membrane, it's a bilayer membrane. And so, as you know, it's hydrophilic, and then hydrophobic, and then hydrophilic. So in this orientation, this is the inside of the cell, this is the outside of the cell, and there's layers of lipid here, hydrophobic. Okay, just real big picture kind of stuff. And if you look, let's focus on, go back to, these are disulfide bonds. You notice this is cysteine 85, this is cysteine 126. Notice that this is cysteine 71, and this is cysteine 46. And notice that this is cysteine 62, and this is, let's see, get a clean shot of it, cysteine 104. Okay, now let's kind of look at what's going on here. And that is, if we look at this disulfide bond, disulfide bonds are very, very, very strong in the world of protein chemistry. Notice that, number one, it's inside the hydrophobic membrane, so it's not going to be accessible necessarily to water. And that is, you can break a disulfide bond with the hydroxyls of water, and so it's going to be very, very, very stable. Now, this disulfide bond, the 7146, is tying this alpha helix into the beta barrel. So if you look big picture, we have an alpha helix here. We have a beta sheet, beta barrel here. Again, this is inside the cell, this is outside the cell. Now let's think about what would happen if we did not have this disulfide bond, which is holding this alpha helix to the beta sheet. Look how long this is. This is quite a big tail. Okay?
And is
this thing would be flopping all over the place.
That's what I was wondering.
Okay? It would be flopping all over the place. But what's really interesting is, guess what? You need it to have extend into the fluid, this is cytosol, so this is outside the cell.
The extracellular fluid?
Exactly.
Okay.
And that's where it binds this. So if this disulfide bond was not here, this would float around everywhere, and you would not necessarily have it oriented exactly. So this is held in, this is more or less holds in the membrane, and then this ties it in. Now, just as a curiosity, imagine that you have 3 disulfide bonds. And so you have to ask yourself, well, how does the protein know to form a 4671 disulfide bond, and instead of, let's say, a 4685 disulfide bond or a 46126 disulfide bonds? So you have to look at all the possibilities of which disulfide bonds would actually be formed.
Interesting.
This is the protein folding problem. One aspect of it. How do you get, of all the possible disulfide bonds, how do you get the right ones to form? Okay, so now you're starting to get an idea of what we have here. We have a beta sheet, and it is in the membrane. And then we have attached to it very strongly is this alpha helix of the receptor. And then this, if you see this, the binding of the blue, which is this semaglutide to the receptor, you can see this is also a very well-known motif of a alpha helix binding to an alpha helix. So let's look at this a little bit further. Let's figure out if these are parallel or anti-parallel. In other words, as we know from peptides, there's a hydroxyl end and a amino end. And let's see, so this would be a 29 and 32, so increasing in numbers. And this is a 10 and increasing. Okay, so these are parallel to each other. All right.
Okay.
And so if
That's
one,
cool.
yeah, isn't it kind of cool? If one wants to do a little bit of research to find out and notice that they're pretty much aligned, and that is they're not at a given angle. Okay. And I think that if you go and do your research, you'll find that there are very definite parallel and anti-parallel structural motifs that reoccur over and over again. Okay, so that's kind of what one has here. Now let's look at the beta barrel a little bit more, and that is we come out here and we have a leucine, lysine, serine, asparagine, serine, proline. Oh, prolines are really cool. Right next to a tryptophan. So proline forces a bend, and trip, arg, asparagine, leucine, serine. Oh, so this, oh wow. Oh, this is cool. Look at this. This is a terminal end. Okay, so this is, if this is 29, this is 128. All right. And remember that the disulfide bond we first looked at, which was the 4671, is holding the alpha helix to the beta sheet. But let's look, and here we see the same, very, very similar thing happening with the disulfide bond of cis 85. And, oh, come on, where are you? in cis 126. Imagine what would happen if this disulfide bond was not here. Then this entire portion of peptide would not be adhered. So let's follow this out a little bit. And what we find is, guess what? This is up here, and we have a mirror down here of this. There again, tying the ends of the protein together. Okay.
So
And then
stabilizing that barrel within the cell membrane.
That's our speculation.
Yeah.
Yeah. You look at it and you go, huh, that's kind of cool.
So it kind of anchors the receptor in the membrane while allowing a portion of it, that alpha helix, to be free enough to associate with the semaglutide. So is our hypothesis the association between the GLP1 receptor, transmembrane receptor, with the semaglutide peptide, that when they associate, that results in a conformational change of the receptor, and that information gets transmitted to the internal. It is the association that triggers the message being sent, like hitting return on your keyboard.
Yes. Or back to Linus Pauling and his Nobel Prize, the lock and key. Yeah.
Even though this isn't an enzyme, right? Because he was talking about an enzyme
Exactly.
reaction, and this is not a reaction. This is just an association.
Yeah.
Because what we find for the semaglutide, well, not for the semaglutide, but for regular native GLP1, it associates, and along comes DPP, and it degrades it, so it quickly stops the signal of the association.
Degrades it.
The semaglutide, it chews it up. But for GLP1 rather, the semaglutide has a built-in DPP4 site that inhibits the enzyme from degrading the semaglutide, so it continues its signal for a long period of time.
So what would be very interesting is to see where the cut site of the semaglutide is. And that is, if the cut site is, let's say, if the cut site is out here somewhere in the extracellular fluid, then one could see you could have it bound and have it easily cut. And it would be harder to imagine where, if it was in this portion and it was bound. But then again, you don't know. You may only be able to cut it when it has a given conformation.
Well, no, I think
and
you're
this
right,
is getting way
because
into speculation.
The DPP4 recognition site is right around amino acid or peptide number 7, 8, right around there is where that site is. So you're right, that would be further out. Now remember,
what's that?
That over here is semaglutide, and it starts with number 7 because,
which is a protein.
Unlike the native GLP1, which starts at peptide 1, semaglutide doesn't start at peptide 1.
Right.
Because the semaglutide has chopped that part off and introduced a DPP4 moiety that prevents it from being degraded by the DPP enzyme. So it is stable. This conformation, this binding, or rather association, is longer lived.
So you can see it's very, very, really beautiful. And that is, you have your beta sheet, which associates with an alpha helix held together with a disulfide bond here, a disulfide bond here, and a disulfide bond here. And then this becomes the extracellular, this becomes the intercellular. And the thing to ponder is the nature of the conformational change which occurs. So, and that's beyond, that's the beyond, yeah, that is
That is a different conversation, but I hear what you're saying.
You can write your thesis on this kind of stuff. Yeah, and where are the waters binding, and where are the salts, and the anions and the cations? Anyway, but that's a whole different thing. But hopefully, and then to recognize that it's kind of interesting that the deactivation bind site, the deactivation cleavage site, is actually on the portion which has the greatest exposure to the extracellular fluid.
And we know that GLP1, the native GLP1, has a half-life of like 1 minute plus or minus a minute. It's very short-lived, so it gets quickly degraded because I guess they don't want that signal when we evolved, that signal wasn't intended to stay there for days. So anyway, all right. Well, thank you.
So to put that in context, how many times a minute does the blood circulate through the body? Because the GLP1 will be carried in the bloodstream, right?
Yeah, it's associated with the albumin.
Right. Okay, that's right. So there's a pool of it. Never mind. All right. Yeah, you're right. Thank you.
I will be looking for the next crystal structure to show the semaglutide bound to the albumin, because I think at the tail that's in solution, that extracellular tail is actually associated with a reasonable chunk of albumin sitting there. So it's
that'll be neat to see that. Yeah, I'm looking forward to that.
Yeah.
So let's take. I've been rotating it here, in the plane of the bilipid membrane. Let's actually look down the helix, and this is kind of fun.
That's beautiful.
You can see that, and then align it to look down here. And by doing that, we discover something very, very interesting that we did not see previously. And it's the following. Notice that here's your beta sheet, okay?
Yes.
And we know that the interior of that is well, we don't know. We don't know because we don't see the side chains. But I would ask whether it's coincidence that for this portion of the alpha helix, the axis of this cylinder is lined with the axis of the cylinder, which is here. Okay?
Oh, interesting, yeah.
But when you look at it, it's outside. It's our speculation is that here's the membrane.
Yeah.
So even though this axis is aligned with this axis, the speculation is it's out of here. So the question becomes, well, what are the side chains doing in the native solution? Which and that's, but you can see where if you just look at one orientation, you can get drawn into a different conclusion or a different observation. Oh, that's kind of neat. Look at that.
What do you see?
That's kind of cool. These disulfide bonds are arranged in almost a perfect triangle. See that?
Mm-hmm.
They are about 120 degrees apart.
Are they in the same plane? No.
No.
Okay.
Which
That's
means
cool.
means that, yeah, but not only that, but it kind of is a stair. Almost a rotational stairway.
You've run into that before, have you?
Yeah, I have. But that's interesting. But no, by sequence it goes down and then it comes up, but by spatial it actually goes, it goes in the same. Oh, that's interesting. It's kind of cool. Yeah, so we have big floppy here and big floppy there. Yeah, so anyway, those are just random thoughts on a structure
that you've never looked at before. I just pulled him in and said, "Hey, you're walking down the hallway. You want to take a look? You're a resident crystallographer."
So those are my thoughts on it.
Thank you very much.
You're welcome.